Okay, thank you for the introduction. I would like to first thanks to the organizers of the International Cancer Research Conference for inviting for this invitation to give a speech about our study. As you can see in the title, our work is on microRNA expression analysis, which revealed biomarker candidates for improved diagnosis and prognostication of pediatric Burkitt lymphoma. Should we change the slides? But the slide didn't change. Okay. So most of the people in cancer research, as you know, is working on carcinomas, which is actually about 90% of all cancers. But different than other groups, we are actually focusing on lymphoid cancers, which constitute around 4% of all cancers. So lymphoid cancers are generally, most of them are lymphomas, but there are also plasma cell disorders and CLR. These cancers derive from B, T, or NK lymphocytes. And the first lymphoma identified is Hodgkin lymphoma. It's called also Hodgkin's disease long time ago. Any kind of lymphoma identified after that is classified as non-Hodgkin lymphoma as a large group. And among these non-Hodgkin lymphomas, as you can see with the red arrow, a Burkitt lymphoma only constitutes 2.4% of all non-Hodgkin lymphomas. So it can be considered as a rare cancer type. So the most, this Burkitt lymphoma can be observed in adults. Also, it can be observed in children. And importantly, it's the most common non-Hodgkin lymphoma in children. And this cancer derives from the neoplastic transformation of centroblasts as the cells, and is characterized with rapid tumor growth, and fast diagnosis for therapeutic intervention is very much important. Here you see the picture, the figure of a spatial structure called germinal centers, in which naive B cells are converted into antibody-secreting plasma cells, and this structure is found in secondary lymphoid tissues such as lymph nodes or tonsils, and it transcends the forms, and it's very much important for if there are important processes happen here, and if there are dysregulations happen, it may give rise to a variety of different kinds of lymphomas, especially B-cell lymphomas, and Burkitt lymphoma also derived from that, as I mentioned, from centroblasts. Now, I would like to briefly mention about the traditional biopsy for the pediatric Burkitt lymphoma. Just a second, please. So, as you know, for carcinomas, the gold standard is the solid biopsy characterization, which is called traditional biopsy. Generally, these biopsies, tumor tissues are taken from lymph node or the bone marrow in the case of Burkitt lymphoma, and the sections are obtained from these biopsies, and morphological characterization is done in the histopathological level. So the characteristics of the cells, centroblasts, the transformed cells need to be identified for the diagnosis. Another important diagnostic methodology is immunophenotyping, and it can be done through different ways. One of the major ways can be done is through immunohistochemistry. The other one is through flow cytometry. As you know, this is based on identification of antigens expressed on the surface of tumor cells or not expressed on the tumor cells to identify the tissue content. Also, molecular cytogenetics, especially fluorescent in situ hybridization, is important for diagnosis of these lymphomas because there's a characteristic translocation for Burkitt lymphoma. Now, one of the important area of research recently in diagnosis of cancer is to figure out a way of avoiding standard biopsy, or at least trying to reduce the importance of standard biopsy. Because standard biopsy is a problematic procedure. It's an invasive procedure. It is time-consuming, and the sampling of the tumor tissue is localized. In other words, there may be multiple tumor tissues in the patient, and then you are just taking a part of one tumor tissue for characterization, and certain genetic and epigenetic aberrations may not be reflected in the part of the tumor tissue that you are trying to characterize. And depending on the anatomical structure, it may be difficult to obtain. It may be small, it may be difficult to reach, and it may be located, the tumor tissue may be located in a dangerous place in a way that when this invasive intervention happens, it may also damage some critical anatomical structures in the patient that may cause more trouble, or there may be other risks, such as infection may happen. So standard biopsy is still the gold standard, and we and other groups are trying to identify liquid biopsy options. This liquid biopsy, as you may have heard, is the biopsy actually based on body fluids, mostly peripheral blood, but it may be urine, saliva, and other kind of body fluids as well. Obtaining these liquids are easier, characterization is faster, and the tumor tissue profiling in an indirect way actually is more comprehensive. You will understand why I'm saying this in the next slides. It's easy to obtain, minimal pain or minimal risk, and it's minimally invasive sometimes, and most of the times actually it's called non-invasive methods. So before actually talking about our study, I would like to briefly mention certain potential clinical applications of microRNAs as biomarkers, because today's topic is about microRNAs. So tumor tissues, as you know, that there are differentially expressed microRNAs that reflect actually tumor biology that may be used for diagnostic purposes for cancer type classification. But even better, if we can take a liquid biopsy, we can take the blood, separate the plasma. There are microRNA biomolecules also in the plasma of the patients that may be potentially used also for differential diagnosis, especially when we want to differentiate between the cancer versus the similar type of other diseases, which is, for example, benign modules. Today's talk is not about therapy, but this kind of biomolecules can be also, of course, used for selection of patients for targeted therapy. For example, some sort of microRNA expression may be associated with the resistance to a specific therapy, or it's also possible to do serial blood collection during treatment in every 2 months or in every month to monitor the response to the treatment and the differences happening in the concentration of the plasma circulating microRNAs may be indication of a relapse that's happening in the cancer patient, or it may also show the disease progression. So it can be used for monitoring purposes. So we mentioned about this circulating microRNAs, and the question is that where does this circulating microRNA come from? So the circulating microRNAs are coming from different sources, but it's all related with the tumor tissue. You know that some tumor tissue, some parts of the tumor tissue may be necrotic, especially in the anoxic conditions when there's no blood supply. The cell may be dying through necrosis, or sometimes even there may be apoptosis, and this, in these cases, the microRNAs will be released into the blood circulation. Also, it's possible that there are microvesicles called exosomes, which are important in the communication of different types of cells in eukaryotes. They may be, so microRNAs can be released within, inside these exosomes, and they are called circulating exosomal microRNAs. So with this background information, the hypothesis of our study was that differentially expressed microRNAs can be used to improve diagnosis and prognostication of pediatric Burkitt lymphoma. And the first aim of the project was to identify microRNAs differentially expressed in tumor tissue and peripheral blood of treatment-naive peripheral blood, pediatric Burkitt lymphoma by using microRNA sequencing. A second aim is to investigate the potential of tumor tissue and circulating microRNAs to improve diagnosis. And the third aim was to examine the relationship between differentially expressed tumor tissue microRNAs and the PBL survival. So this slide is briefly showing the type of workflow we followed during this project. As a reminder, pediatric Burkitt lymphoma is a rare type of cancer, as I said. It's a lymphoma, even not so much frequent, but it's a very aggressive type of cancer. the tumor tissue growth is very fast. So we had number of 17 cases for this study, which is not a very high number, but for a rare cancer type is good enough. And as a control cases, we use obstructive sleeping apnea case, which is a non-cancer group. It's not totally healthy, but very close to healthy, and it's around 22 cases we had. So from PBR patients and this OSA cases, we obtained the peripheral blood, separated the plasma, then used a special kits and isolated the circulating microRNA and circulating exosomal cell-free microRNA. And then we applied microRNA sequencing and identified the microRNAs overexpressed in pediatric Burkitt lymphoma plasma samples. Simultaneously, we also obtained the formalin-fixed paraffin-embedded tumor tissues from the PBR patients. And from these sections, we identified the tumor microRNAs and then performed microRNA sequencing. And as a control of the tumor microRNA from tumor tissues, we also obtained the tonsil from the obstructive sleeping apnea cases, and then generated single-cell suspension, did immunophenotyping, then isolated the centroblast through FACS, and then from centroblast, we isolated centroblast microRNA, and we did microRNA sequencing. So we compared to circulating microRNA and exosomal microRNA in PBR versus the control, and the tumor microRNA expression pattern in PBR patients versus the tonsil centroblast microRNA from the control cases. But before showing the results related with this study, and this is a recently published study actually today, what I'm talking about, we also did another analysis. We checked the total microRNA concentration. So this was before the microRNA sequencing, actually with the Agilent bioanalyzer, the microRNA concentrations were obtained. And doing a simple calculation, we calculated the per ml concentration of the total microRNAs in the cancer group, Burkitt lymphoma versus the control group. As you can see in panel A, the circulating cell-free microRNA concentration is higher in PBR case compared to control cases. As you can see in panel B, there was not a statistical significant difference between the PBR versus the control cases. So circulating cell-free microRNAs were higher, but circulating exosomal microRNAs were not higher in PBR. As you know, people who are working on these diagnostic studies are generally familiar with the receiver operating characteristic curve analysis, where we identify the area under the curve values. If it is more than 0.5, it is generally good enough. If it is close to 1, it is even better. So when we look at the total circulating cell-free microRNA concentrations, we observe the very statistical significant association, the diagnostic potential of total circulating cell-free microRNA concentrations with the AUC value of 0.81. As you can see in panel B, we had the AUC value of 0.65 with exosomal microRNA concentrations, and these are plasma exosomal microRNA concentrations, but it was not statistically significant. So total circulating cell-free microRNA concentrations has the potential to improve PBR diagnosis. Now, I don't want to get into much detail about the bioinformatic pipeline we use for the microRNA sequencing data analysis to identify differentially expressed genes. But basically, I just want to show this slide to give a brief idea about what kind of workflow we use. So PBR tumor tissue, tonsil centroblast, and also PBR versus OSA circulating cell-free or exosomal microRNA. These were the comparisons. We obtained the raw next generation sequencing rates after the microRNA sequencing, and using different modules of the MIR-DEEP2 program, we aligned the NGS rates to the human genome, then identified microRNAs, then identified the expression levels of the microRNAs, and then using the Agile program, we identified differentially expressed microRNAs in pediatric Burkitt lymphoma samples. We also use a function of Agile program. As you can see, the microRNA levels normalized as CPM values here. Actually, I would like to show with the pointer. So can you see the pointer now? Maybe it's better I show this way. So anyway, you can see that in this slide, we took the most upregulated microRNAs and most downregulated microRNAs and performed hierarchical clustering of pediatric Burkitt lymphoma tumor tissues and OSA tonsil centroblasts. So as you can see that when we use 10 highly upregulated microRNAs, they are upregulated in PBL, or the 10 most significantly downregulated ones, we can totally distinguish the pediatric Burkitt lymphoma tumor tissue samples from the tonsil centroblasts. And if we use only 5 microRNAs, which is upregulated, or 5 microRNA, which is downregulated, it's actually the other way around in PBL cases, we can still totally distinguish the Burkitt lymphoma versus the cancer cases. Burkitt lymphoma versus the non-cancer control samples. In order to be more sure about actually our pipeline, sure about our results, especially with the tumor tissue microRNA, we also use independent data set. So here, what you see is that from our in-house data, as I mentioned, we had 17 cases. We using Agile program, we identified microRNA significantly differentially regulating PBL tumor tissues. And then also we obtained another microRNA sequencing sample set from NIH database. It is Burkitt lymphoma genome sequencing project. And then we compared with our in-house OSA tonsil centroblast samples, and with the use of Agile program, actually we identified microRNAs significantly upregulate or downregulate in PBL tumor tissues. So the aim here is to identify a more accurate set of microRNAs differentially regulated in tumor tissues by comparing our in-house PBL cases microRNA data versus the independent data set data, and generated the Venn diagrams and identified with the microRNAs that are upregulated based on both data sets, or downregulated based on both data sets. So here you see the results. Based on the in panel A, in our in-house data, when we compare with VGLSP independent data set, there's 168 microRNA upregulated compared to control cases, and 128 microRNAs downregulated in both data sets. So here you see the using this 168 plus 128, total 396 microRNAs differentially expressed based on both data sets, we identified the certain pathways that are related to cancer, such as to be significantly enriched in this microRNA list. So the common list of differentially expressed genes, as expected, were enriching cancer-related genes. This has an implication, of course, in the role for the role of these microRNAs in Burkitt lymphoma development. But our main purpose in this study was not to characterize the specific microRNAs taking part in the development of PBL, but we wanted to see what kind of potential biomarkers we can identify. So what we generally focus on the ROC curves, as I mentioned, which is used for this kind of analysis. From the in-house data, we also compared the ROC curves from the in-house and validation data sets. So here you see the workflow, microRNA sequencing count data of our in-house series cohort, the CPM values count per million obtained, and we identified top 25 significantly upregulated microRNA. So these are details, but what we did is that using an E0 program, we identified the AOC curves actually. But here also from the independent data set, which has actually much more sample here, 88 PBL cases, we also have the microRNA set data. So get the CPM values here, and with E0, we generate the ROC curves. So these ROC curves are double ROC curves of the microRNAs that are most significantly upregulate in our in-house samples, but also, as you can see here, these AUC values also in the BLGSP dataset, as you can see, the top 10 upregulate microRNAs in the in-house and independent cohorts is close to 1, such as HSA MIR 126, HSA MIR 100. You can see that in both datasets, the AUC values were almost close to 1, which is almost perfect. It means that the sensitivity is almost 1. Specificity of almost 1 can be achieved at certain levels. But of course, for some of them, this was not so much perfect. But for most of the microRNAs we identified to be upregulated in our in-house data set, it was also validated to have diagnostic potential in the independent data set. So we also did survival analysis, generated Kaplan-Meier plots with tumor tissue microRNAs that we identified in our in-house, not in-house actually, the validation data set, because the in-house number was a little bit low. So we identified 168 microRNAs that were 168 microRNAs that were overexpressed in both our in-house and independent data set, and 133 overexpressed microRNAs had sufficient survival data. This is from the independent data set had this information. So we used that data set to associate with the survival. And 7 of these 133 overexpressed microRNAs were significantly associated with shorter overall survival in PBR cases. So they are not only overexpressed in general in PBR cases, but the case which has relatively higher expression has shorter overall survival. And by contrast, 4 of the 128 underexpressed microRNA, these are underexpressed in PBR cases, were associated with, again, poor overall survival. In this case, they are not only underexpressed as a group general in PBR cases compared to controls, but the ones that has lower expression was associated with poor overall survival. As you can see here, the overall survival probability, the panels A to G is actually showing the overexpressed microRNAs that are associated with whose higher expressions are associated with poor survival. So red curves are showing high expression, the case with high expression. As you can see that there are certain microRNAs, as I mentioned in the previous slide, is associated with poorer overall survival probability. Also, as you can see from panel H to panel K, the certain microRNAs that are underexpressed, not only underexpressed, but their lower expression levels in those generally underexpressed cases was associated with poorer overall survival in PBR. So we identify several microRNAs that are associated with the prognosis of PBR as well. Now I talk about tumor tissue microRNAs. Actually, as I mentioned at the beginning of the presentation today, that our main aim was actually to identify circulating microRNAs that can improve the diagnosis of PBR cases. So as you can see in this slide, we generate also ROC curves using circulating cell-free microRNAs overexpressed in pediatric Burkitt lymphoma cases. As you can see between panel A and panel D, even single microRNAs can reach to AUC value of 0.74. Remember that anything actually more than 0.5 can be considered to have some kind of a diagnostic value, although it's not so much high enough. The closer to 1, it will be better, but this was still significant. And we didn't reach very high AUC values, but it was still probably important, and they were also generally statistically significant. But when we do combine 2 by 2 these microRNAs, these are plasma microRNAs circulating cell-free. As you can see, we obtained better results. AUC values reached past 0.8 for some combinations. We did the same analysis with the exosomal circulating cell-free microRNAs overexpressed in the PBR cases. As you can see, again, when we only check 1 by 1, the microRNAs overexpressed, the AUC values were a little bit better, but it was not still so much high, 0.77 for this one, 0.76 or 0.75. So generally it was between 0.7 to 0.8. So this same thing we did also for the exosomal microRNAs. We combined 2 by 2 or 3 by in triple groups actually, to see whether we can improve the diagnostic potential of these microRNAs for potential liquid biopsy. As you can see, when we combine, when we analyze together 2 microRNAs, we can reach the AUC values better, 0.91, and it's very much statistically significant, and 2 by 2, 0.88, 0.87. If we combine 3 of them together, as you can see between panel G and panel I, we can even reach to 0.97 or 0.93, which is almost similar to the level we reach with the tumor tissue microRNAs, which are overexpressed also in PBR cases. So as a proof of the principle, we did cross-validations of the expression of selected microRNAs with QRT-PCR. So here actually in between panel A to panel C, what you see is the tumor tissue microRNAs, 3 of them here. And the y-axis is showing the QRT-PCR expression levels, and the x-axis is showing the microRNA sequencing-based expression levels. As you can see, the Pearson-R correlation is 0.94 between microRNA sequencing and the QRT-PCR for MIR125-5P, and it's also 0.91. P-value is quite significant for another microRNA, MIR105P, and this one is also good, but these values were a little bit low when we compared to microRNA-seq and the QRT-PCR values, but there was still obvious and significant positive correlation for the circulating microRNAs. Here, the panel D is showing the circulating cell-free microRNA correlation here is close to 0.5, statistically significant. Panel E and F is showing actually 2 microRNAs the expression comparisons of 2 microRNAs, and the Pearson value was 0.68 for one of them, the other one 0.48. So in general, we were able to successfully validate the microRNA sequencing results for selected microRNAs by using QRT-PCR. So in conclusion, total circulating cell-free microRNA concentrations can be used to improve non-invasive diagnosis. MicroRNAs overexpressed in tumor tissues of PBI cases also have strong diagnostic biomarker potential. But as I mentioned, if we analyze the microRNAs 2 by 2 or 3 by 3 for especially circulating exosomal cell-free microRNAs, we can also reach to very high diagnostic potential. So prognostically significant differentially expressed microRNAs may be also useful to guide the patient management and therapy. So before finishing my presentation, I would like to thank our current and previous lab members for contribution to this project and other projects. I would like to also thank TÜBİTAK 101 for funding specifically this project, and other funding agencies that provide financial support for our ongoing or past projects in our lab. I would like to also thank several investigators who contribute, especially with the cases from Hacettepe University, Dokuz Eylul University, Ege University, Health Science University, and Mansi University in Turkey. So I think it's my last slide. Yes, thanks for listening to me. I was the last presenter, I think.